What we're going to be discussing today is nutrition, the microbiome, and immune balance. And if you think about it, there is a nexus. These vectors are coming together in a way that is giving us an understanding of how they affect each other. And I think that that is extremely important in being able to really understand where we are going. And to begin, excuse me, I'm just going to cough. To begin, I really would like to be able to give you an understanding of where we have been. And in order to know that, I wanted to talk to you about the very famous tree model. And I know that all of you are aware of this. If not, you know, this is the classic model that is used in integrative medicine and many other scientific areas. And it really discusses the model which is exemplified by a tree. And what is the tree? You see the leaves, you see everything out there. Those are the diseases. But of course, what we are really looking for, or should be looking for, are what are the root causes. Excuse the pun, the root. We want to know what are those root causes. And as Mark Hyman has mentioned, and many others, and it's just lately that he has discussed this again, is we really want to understand the problem of conventional medical thinking. We do know that conventional medicine thinks and focuses on symptoms, doesn't focus on the systems themselves. It focuses on organs. It doesn't focus on the organism. Again, all these things we know, but it has to be repeated. The disease you have is what they concentrate on, and not why you have the disease. And so they're not focusing on the root causes. And that's something we would like to look at today, because in doing that, we'll be able to get an understanding of what some of these major causes of disease are. And we know what most of those are. We know that it involves toxins, allergens, stress, and that's something stress we'll talk about in much more detail over the next number of weeks. And we know about the microbes, about the microbiome, about diet, especially about poor nutrition. And this is what we're going to be going into in detail over the next hour or so. And we also know what the ingredients for health are. And I know that at FDN you stress this with your DRESS system. And we know that if we can decrease the amount of processed foods, if we can balance the nutrients and stabilize the environment, we have a very good way of dealing with it. And something which I will discuss today, which is not discussed enough, is sufficient exercise and sleep. And we will discuss sleep in a whole number of contexts as we move through this hour. I don't know how many of you are familiar with the term blue zones, but for those who are not, I think it bears repeating. The blue zones have been known traditionally as the geographic areas where they have found that people live longer, they have much lower rates of chronic diseases, etc. And it goes from places like Loma Linda to Okinawa in Japan and many other areas throughout the world. And the hallmarks of what you're looking at when you actually see what's going on there is that almost all of these have plant-based diets, they have regular exercise, there's alcohol very much in moderation, they get sufficient sleep, they get at least the 3 and 4 cycles of sleep, and they have very stable social networks. So what are we talking about when we begin to discuss this? Well, one of the things that we need to understand is autoimmune epidemic, because the antithesis of the blue zones is what we are seeing today, definitely in North America. And that is what one could really describe as an epidemic, because we know that 1 of 2, maybe even greater amounts of Americans are suffering from some kind of immune disorder. It's actually quite a depressing thing to look at, because you're seeing a tremendous amount of these diseases increasing in the US, much less in the rest of the world, but unfortunately they're beginning to catch up. So approximately 10% of the world population is suffering from what we're calling AI, and that's not artificial intelligence here, that is autoimmune diseases and disorders. So we need to ask ourselves, why is this happening? One of the things, for example, is autism spectrum disorder, or ASD, in the US. And for those of you who have not seen this particular graph, take a look at it. We are not looking at what happened a couple of centuries ago. We're looking at 1975, when 1 in 5,000 people had some kind of diagnosis of being on the spectrum. Now, you can argue if it was 1 in 5,000 or 1 in 4,000, but it was definitely a small number. By 2012, that had gone to 1 in 88. And that has now increased, by the time we have reached close to 2020, of something which we estimate to be 1 in 54. I would submit to you, we've got a problem here. And the question is, why that problem? And by the way, I don't even want to get into the issue of the various types of pesticides and glyphosate and so on, but it's interesting that that curve follows the autism spectrum curve almost right through the curve completely. You have to ask yourself, what is going on here? Well, I mentioned to you that we would be dealing with issues that are really coming together right as we speak. And hot off the press is, what is the effect of the microbiome and autism spectrum? Well, we know that apart from genetic factors, the gut microbiota are now being seen to play an extremely important role in autism spectrum. In fact, children with ASD have a very distinctive and underdeveloped range of their gut microbiota. And it is not related to diet. So we know that there is something going on, and we will discuss what those things are in a few minutes. But definitely, you're seeing with them a very different population within their microbiome. And the microbiome functionalities in this case are associated with their neurotransmitter synthesis. So you're getting a lot less synthesis of neurotransmitters, very markedly reduced, in children with ASD. And that, we know, would have a profound consequence on psychiatric abnormalities in those children with ASD. So what is the conclusion? Well, the conclusion by this group in 2021, I told you I'd give you stuff right off the press. So in 2021, what are they saying? The shifts in, I'm going to read it, the shifts in gut microbiota during early life development, and remember the early life portion, keep that in your minds for the next few minutes. That development may have important functional roles in the pathogenesis of autism spectrum disorder. That's a big statement. And there are many other studies. We could go on forever just looking at this. Here is one which deals with the variation of the toxins that are produced by bacteria in the microbiome and how that is contributing to autoimmunity. This is a study from 2016. And the study is a very interesting one because what they looked at was the gut microbiome development in over 200 infants in Finland, in Estonia, and in Russia, with those that are predisposed to developing type 1 diabetes. And so they follow these kids from birth to the age of 3. And what do they see? Well, we know that early onset autoimmune disease is very common in Finland and in Estonia, and much less prevalent in Russia. And the results are interesting because what they show that in those that develop type 1 diabetes, it's preceded by a drop in microbial diversity. There's another term, diversity. I want you to keep that in mind. So there is a drop in the diversity of the microbiome, and it's disproportional decrease in the number of species that are known to promote gut health. So within the microbiome, there is a loss of the species of bacteria which are known to promote gut health. In Russia, Bifido and E. coli and the LPS predominates. Well, you'd say, well, wait a minute, those are toxins. But yes, they are toxins, but they are very potent activators of the innate immune system. There's another term I want you to remember because we're going to go into that in more detail. In Finland and Estonia, what you see is a predominance of Bacteroides and its LPS. Well, guess what? It inhibits the innate immune signaling. And so therefore, what you're getting is early colonization by microbiota in let's say, Finland and Estonia that are silencing or they're decreasing the immune response. And that may prevent what is called immune education in the child to begin to learn at an early age what it needs to do to fight all of the different infections, and therefore will be enhancing possible autoimmune disease later in life. And that is part of what we call the hygiene hypothesis. What's the hygiene hypothesis, you may ask? Well, we know that since the middle of the last century, we have had an incredible decrease in infectious diseases. We've really almost conquered those diseases. Rheumatic fever, hepatitis, measles, you name it. But guess what's happened at the same time? On the left, you see autoimmune epidemics of asthma, type 1 diabetes, Crohn's disease, multiple sclerosis. We could go on forever. And that takes place mostly in industrial countries. So you're getting this incredible increase in autoimmune disease in industrialized nations, and it is explained in whole or in part by changes in the early microbial exposure, which leads to altered immune maturation, like you saw in that previous study. So if we were to look at the pathogenesis of chronic inflammatory disease, what are we looking at? Well, the first thing is genetic predisposition. Of course, you can't forget that. Genetics plays a role. But let me be very clear. Genetics doesn't change over a year, over 100 years, 1,000 years. It takes tens to hundreds of thousands of years. So that cannot be the major reason why we are seeing increases in chronic inflammatory disease. One of them is environmental triggers. What is happening in our environment? So of course, nutrition, which we're going to go into in a lot of detail in the next few minutes. Infection, antibiotics, toxic products, all of that together with the microbiome, especially when it's not functioning properly, when it becomes dysbiotic. That leads to dysregulated or imbalanced immune response, which then makes inflammatory problems even more problematic. And that, together with the increased intestinal permeability and the loss of a barrier integrity due to a great degree, dysbiotic behavior, is going to lead to inflammatory disease. And the question then becomes, what are the clinical outcomes after that? Are we going to get resolution of the problem? Are you going to get chronic inflammatory disease? Or is it going to get even worse? And are you going to end up with an autoimmune problem? So when you look at chronic inflammatory disease, I want to pose for you a theoretical construct. And that is the whole issue of what inflammation really means, because it underlies most metabolic dysfunctions. And we know that it is caused by an imbalance in the immune system. Now, what does that mean? Well, we'll tell you in just a few minutes. But the goal, the goal is to create immune or immunobalance. And how do you do that? Through immunoregeneration. These are big words, but let's try to dissect this a little bit. In our modern societies, this war is not being won, folks. And Carl mentioned that earlier. And why not? Because the management approach is not sufficiently targeted. The health aging concepts are not being followed. There's a deficient exercise. We have an unbalanced diet. There's industrialized food production, to say the least. So the result is a plethora of inflammatory states: obesity, type 2 diabetes, inflammatory bowel disease, IBD, rheumatoid arthritis, cancer, etc. And so what you need to understand is that there is something which we're beginning to now discover in much more detail. And that is something that we call immunosenescence, otherwise known as inflammaging. As we age, we change the way in which we can handle inflammation. In fact, what we're beginning to understand is that some cells enter into a chronic inflammatory state. New immune cells, we know, are created every 1 to 2 months, constantly turning over. But some of those immune cells are carrying the history of the injuries that have happened earlier in life. And they create what we now know to be mutating or mutated stem cells. In fact, we've given them a name. They're called CHIP cells. These are clonal hematopoietic indeterminate potential cells. In other words, these cells remember what has happened to them. And when the new cells come along, they already have that history. And guess what? As you age, you get more and more of those cells surviving. By the age of 65, 10 or 15%. By the age of 80 and over, fully 30% of those cells are, in fact, CHIP cells. Now, the body has a mechanism to deal with that. And that's called autophagy or autophagy, whichever way you'd like to say that. That's a well-known concept. In other words, the body is going to eat up those cells. And we're getting a bit of an echo, so if somebody's, there we go, we're off. So the body cleanses damaged cells constantly to renew them with newer, healthier cells. So what's the game? The game is to make sure that repair is more robust than damage. That's the game you're playing. And what is the major autophagy control? Well, we know what it is. It's diet. And so you get the concept, which has been around for a while, but I want to stress it. Diet as medicine. Why? It allows time for the immune system to reset. This whole idea of intermittent fasting, which we'll go into in a few minutes. Enhancing the intake of phytonutrients. Ridding the body of toxins. Nothing that you don't know, but we're now putting it in a conceptual construct that'll give you a better idea of what's going on. Get rid of those endotoxins. Get rid of the parabens, the phthalates, bisphenol A, etc. So let's spend 2 minutes to remind ourselves of one part of this diagram, and that is increased intestinal permeability. You all know about gut-associated lymphoid tissue, or GALT. If you don't know it, it's extremely important. It is part of what maintains GI, or gastrointestinal mucosal immune hemostasis. It keeps stuff in a hemostatic force. Here is a mouse cross-section. This is an intestinal area of a mouse. And what you see are the epithelial cells, the one cell layer, and sitting here is a Peyer's or Peyer's patch. And what is that? That is where the immune cells are created and sit around, and they are waiting to do their job. That is what's affectionately called the second brain. This is where you have the dendritic cells, the B cells, the T cells. There they are sitting and they are interacting with the epithelial cells, the enterocytes that are this one cell wonder that keeps the tube which enters our body that we call the gastrointestinal tube from allowing to bring in nutrients and bacteria and not destroy our bodies at the same time. And part of that gut-associated lymphoid tissue is as a containment system, because together with commensal bacteria, the good bacteria, that is what is able to keep potential harmful antigens, food antigens, you name it, from entering into the systemic circulation and causing problems. And what is one that I would like to stress today, and that is secretory IgA, one of the immunoglobulins that sits within the area of the lumen of the gut. And it is the first line of defense. It protects the intestinal epithelium from various pathogens, from toxins, enterotoxins, exotoxins. So it is there to clear toxins away, blocking their access to the epithelium of the intestine, let alone allowing those pathogens to get inside the body. It entraps all these different pathogens in the mucus. That's where SIgA lives. It lives in the mucoid layers just above the enterocytes, that epithelium, and together with the mucus, the SIgA glomps on to those pathogens and removes them from the body through peristalsis. It's a beautiful system. But between these epithelial cells, we do have the paracellular junctions, the tight junctions, which now are very well known, and we know that they are controlled by zonulin. And zonulin is the game changer. It is the objective biomarker which was discovered a little under 20 years ago by Alessio Fasano and his group in Maryland at the time. And it is what controls the tight junctions. It's a defense mechanism. Zonulin and all of the tight junctions were really developed to do one thing very well, to flush out microorganisms that manage to get through into the body. You needed to get them out, so you'd fill them with water and shoot them back out into the gastrointestinal lumen, and that's what it did. But of course, if you open a door, not only does stuff flush out, but other things can get in. And so that's where you can end up with a lot of the problems. And so this is a busy slide that is extremely important, because it tells you what happens when you lose this mucosal hemostasis. Well, on the left side, you see number one, that is our normal, physiologically controlled permeability. What do we have? We have cells that are functioning well. They've got tight junctions that are closed for the most part. Beyond that, you have a number of different cells of the immune system. And you have dendritic cells, the macrophages, which eat up and take away pathogens. And all of that is made to be able to deal with what we call anergy. And what is anergy? Anergy is that there is no major immune response to antigens. Everything is quiet. The lumen has whatever pathogens it has, it's being taken care of. Very little is getting through, and the immune system says, thank you very much. I don't have to do anything right now. It's functioning well. But that changes, and it changes on a constant basis. Number two says, what happens if you have some gut dysbiosis, which will cause some zonulin release? It can lead to some minor gut barrier defects. You can begin to open up those tight junctions a little bit. SIgA is maybe not working as well as it should. There are metabolites of the microbiota that are getting too close to these intestinal cells. The mucus is beginning to really decrease and lose the type of mucus that you would normally want. It's not as broad. It's not as as thick as it normally is, this will begin to promote innate inflammatory effects. And you begin to therefore get a vicious cycle that can occur with increasing gut permeability, leading to even more inflammation. Innate inflammatory issues can become what we call a adapted type of immune response, and that can set off a major problem where you now have very large openings between the cells. You get a break in the tolerance. What normally was tolerant now is beginning to create tissue damage, and you enter into a state of chronic inflammation. And that is what you want to avoid. So there are many different types of immediate and delayed intestinal inflammation. And I'm just going to show you this just to give you an idea of all the things that can be happening in the gastrointestinal system: IBD, IBS, diarrhea, the various metabolic problems, diabetes, obesity, metabolic syndrome. People always say, what is that? Well, it's abdominal obesity. You have high levels of blood pressure. The fasting glucose is high. You have high levels of triglycerides. None of that bodes well. You have that in the brain, in the nervous system. I'm going to tell you a little something. Zonulin can also open up spaces between the cells of the blood-brain barrier. We used to think that could never open, but it does. It's a different species of zonulin. So that, in effect, can begin to allow various types of antigens to get into the brain. By the way, even 10 and 20 years ago, we didn't think that was possible. We do know it is now. And etc. Autoimmune diseases, respiratory issues, skin, heart problems, the adrenals, and of course, the thyroid. For those who are trying to write quickly, I'm going to be offering this as a PDF, as a gift to everyone who is on. Thank you. I will clap as well. So we're going to give you this, and you'll be able to look at that at your heart's content.
Thank you.
And so let's take a look just very quickly at some of these issues. Let's take a look at some diseases and disorders to give you an idea of what this means. What is intestinal permeability, and how does that factor into chronic inflammation and autoimmune disease? Well, the incidence of celiac disease is going up exponentially. This is not normal, folks. Since 1950 up to 2020, we've had an incidence of over 7.5% in the last 30 years in the industrialized world. And by the way, it's highest in females and children, and we can discuss that later. If you pool that with the global preference, it's 1.4%, which tells you something interesting again, that the rest of the world has not suffered these issues as much. Why not? That is staring at us as an issue. We need to come to terms with that. And fully, 83% of that is undiagnosed, or it's misdiagnosed. So guess what? It's even worse. Why? Why is that happening? Remember, I told you it's not genetic. It can't be genetic. It's happening too quickly. And why do we feel that it's happening even more in North America than it's happening in Europe? Part of that is the way in which we are utilizing industrial systems to break down our peptides in the production of things like wheat that we use in bread. We do it in a different way than you do it in Europe. People who suffer from gluten problems in North America will tell you they don't have the same problems in many European countries. We know that part of that is because in Europe they use many more steps to break down gluten. And of course, pesticide use. I don't even want to get into that. So we know that celiac disease is caused by an increase in zonulin and a development, therefore, of an increased gut permeability. That's old news, by the way. Very old news. That's 15 years or more. And we know why it's happening. It's happening because of a microbiome imbalance. It's dysbiosis of the bacteria, SIBO, the fungal changes, Candida, for example, parasitic changes, and gliadin in the diet from gluten. That's the active component, alpha-gliadin. And by the way, one of the reasons is because the body perceives alpha-gliadin in the same way as it's perceiving a pathogenic organism, and it acts accordingly. We even have that issue with non-celiac gluten sensitivity. This is a beautiful study by Barbaro and his team in 2015, 6 years ago, in which they were able to show that controls and IBS, that's people who do not have frank bowel disease, but they think they do, but it's coming from another source. They are not having the same increased levels of zonulin that you're seeing with people who have true problems of celiac disease or non-celiac sensitivity, because that's taking place in the intestine, whereas it's not happening for those with normal intestinal issues or IBS. What about diabetes? Well, we now know that one of the major factors, one of the major vectors of what we call type 1 diabetes is, in fact, zonulin and what is happening in the gut. And we know that many of these people, including their relatives, how interesting, there is that genetic component that's raising its head, telling you, yes, genetics probably does play a role. Not the major role, but it plays a role. I don't want to negate genetics. I don't want anybody to think that that is not an issue, but it is a relatively minor issue. Inflammatory bowel disease, Crohn's, ulcerative colitis, etc. Well, we've now known for a while that increased intestinal permeability is central to the pathogenesis of IBD. And we know that IBD has a deficiency of three major components of innate immunity. Remember, the innate system is what we have from our ancestors hundreds of thousands, several million years ago. It's the macrophages, the neutrophils. They were there when we were hunter-gatherers. So you have the physical barriers, the tight junctions. We develop antimicrobial peptides. Both the bacteria and cells within the intestinal wall are producing defensins. Those are antimicrobial peptides. And of course, the cytokines and the inflammatory mediators that say to the macrophages and the mast cells sitting right in intimate contact in the intestine, I think it's time, guys, that you started increasing your number and starting to work, because there's a problem. We got to get rid of those pathogens. So that can lead to inappropriate access to antigens that are coming in the body. And it can change the way in which that second brain, that immune system in the intestine actually functions. So by the way, we know that if you have asymptomatic people with Crohn's disease, they have Crohn's disease, but they are not symptomatic. We know that 1 year prior to them developing exacerbation, they are already beginning to develop increased intestinal permeability. That's well known, study after study. So we know, therefore, that intestinal permeability is an early development in the disease exacerbation. But remember, it starts as an innate immune problem, but as it goes on, adaptive immunity takes over. It gets even worse because the whole systemic issue takes over, and the body begins in various organs to respond to what was, it was in Las Vegas, but now it ain't in Las Vegas anymore. Now the adaptive immune response has made it an overall systemic problem. And how do gut bacteria come into this? Well, here's new information. Again, 2021, the gut bacteria are playing a major role because there are gut microbiota that produce succinate. And succinate, we've known for a while, can produce inflammation. These are inflammation-inducing molecules. They're secreted by the gut bacteria. The uptake are elevated by the macrophages, and that perpetuates the pro-inflammatory state. And we find that tremendously elevated in the serum and the feces of people with IBD. So what's the conclusion of that? IBD is partially driven by changes in gut bacterial activity and by the accumulation of succinate in the gut. And that's one of the things that can lead to chronic inflammation. Is it the only thing? No. But is it now being considered as a very important vector? You bet. Multiple sclerosis. We do know that serum zonulin can differentiate, and you see it in different types of multiple sclerosis. It's elevated in 30% of those with relapsing remitting and so on, and secondary progressive. We know that there's a twofold increase of that in patients with these exacerbated state of multiple sclerosis. And when you're in remission, your zonulin levels decrease to what they were with controls. Same with asthma. We now know that zonulin is increased with asthma as it increases in severity. And so we now know that you can actually look at that as a possible way, a potential way of being able to have a prognosis of where these people are who have asthma, where are they in their severity state. So it may be an additional alternative approach. And by the way, this again is new news. It's coming right hot off the press. I can't let this one go without obesity. You take a look at obesity in all of the industrialized nations, and it's just going up and up. In fact, 45% in 1970 of Americans were overweight or grossly or morbidly overweight, and that has now increased to 74%. And this is not a typo, it is up to 74%. And what do you see with zonulin? Zonulin is increased in all of these cases. There are a whole number of recent studies. They just go on and on and on. This issue is now front and center. It is the effect of these various types of biomarkers and how that can affect it. In fact, some of the new news, again, recently, changes in serum zonulin in individuals who have morbid overweight problems, who have gone through weight loss interventions. What do we see? Well, serum zonulin is markedly reduced after various types of conservative interventions, and it is further reduced after you have what's called bariatric surgery, where part of the intestinal tract is removed. And those reductions in serum zonulin are associated with improvements in diarrhea, etc. So the comment made by these authors is that zonulin is associated with, big surprise, diet, gut microbial dysbiosis, diarrhea, and several other biomarkers of metabolic disease. So what we're looking at is a very important aspect of this. Let me jump to the microbiome and dysbiosis. And just for the fun of it, I just want to go through a couple of minutes to tell you about the gut microbiome. We know that the cells of the microbiome outnumber humans 10 to 1. There are about 150 trillion. The amount of genes, the amount of genetic material in these bacteria outnumber humans 150 to 1. There are 3.3 million bacterial genes versus a paltry 20 to 25,000 human genes. Bacterial species, we know of at least 1,000 or more, and that's increasing by the week. And about 50% or more of the colonic content, the content of the colon and what it lets out of the anal cavity, is bacteria. So we know by weight, we got about 2 or 3 kilograms of bacteria in our bodies at any time. The volume, about 1.5 liters. 1.5 liters. You're not reading that wrong. The species, as I said, about 1,000 or more, and we keep discovering more. The amount of gas that we produce, about 1 to 4 liters a day. We're constantly producing it. And the waste, about 60% by weight. What do we know about the microbiome? Because there's a tremendous amount we don't know. It develops early in life. And please keep this in mind. It is established by the age of 3. And what it's doing is it's expanding the genetic and the functional capacity of the human host. This is extremely important. Because remember, we've only got 20 or 25,000 genes. How are we doing all these things? So in childhood, the founding microbial gut population is shaping our immune function and what is going to happen to us later in life. It's established in the first 1,000 days. And there is a very strong association of early childhood microbial colonization and our health status after that. It's all baked in. And antibiotics can cause a very rapid and dramatic alteration in all of that. So as you may know, to date, 50% of females receive antibiotics during pregnancy. And in fact, we know, this is old news, there's 7 to 10 courses of antibiotics that are given to children by the time they reach the age of 3. Or more, by the way, or more than 7 to 10. And we know that antibiotics and its use parallels obesity. 50% obesity rate in children who are given antibiotics in the first 6 months of life. That's a massive amount. And the subtherapeutic antibiotic treatments in animals that we're going to go into in a minute can change the human microbiome. And so we know that one of the things that I mentioned to you earlier is that if you have reduced bacterial diversity, that is a major element in dysbiotic behavior. And it is going to lead to all of these issues that you see here on the left, from diabetes to obesity, various malignancies, IBD and IBS, of course, something you see in children and babies, which is necrotizing enterocolitis, etc. And the contributors to dysbiosis, we know them well, but we need to keep repeating it. The genetics, of course, never to be forgotten. Our lifestyles, extremely important. The diet, exercise, stress, early colonization, which we're going to go into in more detail. What is the birth mode? How did that child come out of the mother? And the altered exposures to microbes. And of course, medical treatments, antibiotics, the various hygienic things. That is all going to, these are vectors leading to dysbiotic behavior and can change the microbiome and alter intestinal permeability. And so gut dysbiosis has both a local effect, which is necrotizing enterocolitis, IBD, IBS, but also tremendous effects systemically due to, as I said, the metabolites, secondary bile acids, the small chain fatty acids, the vitamins that are produced, the inflammatory cytokines which are produced by bacteria, the various types of toxins. These are the pathogen-associated molecular pattern molecules. That's a big term, PAMPs, LPS, peptoglycans, etc., all produced by microbes. What do? They create this cornucopia of plenty of diseases, of which the dysbiotic behavior is a major factor. Is it the only factor? Of course not, but it's a major factor. I don't know how many of you know about this, but starting in 2007, there was a project of NIH which was called the Human Microbiome Project, and it continues to this day. And it talks about the DNA of microbial communities in different body parts. So it's not just different people or different microbes. Within the same person, you have microbes in different parts of the body that are living on different planets relative to each other. So each one of the points here, by the way, represents one microbial community in one body site in a healthy subject. So if you take a look at what you see in the oral area, you could think of them as microbes living in the sea. And that is totally different from microbes in the same person, remember, with the microbes that they have in the feces. Or for that matter, the ones they have in the skin. So we know that humans are DNA identical about 99.5% to other humans, or even more than 99.5%. What they don't share, and don't even share within their own body, are the same microbes. They only share about 10% of the DNA in similarity of gut microbes from one person to the next, and for that matter, of the same body areas. For example, we know that if you look at the feces of two individuals, they're going to have a very, very different types of microbial population. And we know that if you take a look at the diversity of animals in the wild, that diversity is quite stark. There's a tremendous diversity in those animals. Put them in cages in a zoo, that divergence decreases dramatically. So what about non-Western humans living in different parts of the world? Their diversity is even worse than that of the animals living in the cages. But it doesn't stop there. What about the diversity of humans living in industrialized society, let's say the US? It's not just the US, I'm not singling out the US, but definitely they are the poster child for that. That diversity has decreased dramatically. We don't have that diversity anymore, folks, and that is causing us tremendous trouble. So let's take a look at two things. Let's look at birth mode and maternal flora. By the way, we could look at all of these things. But let's take a look at one study in particular. And that's the birth delivery mode and the microbiome. What happens to the microbiome depending on how the child came out of the mother? And it's called the baby biome study. It's been going on for quite a while. This was published in Nature in 2019. By the way, everything I'm telling you is not a figment of my imagination. This is all based on not just one study, numerous studies. I'm just giving you one here. So a whole 1,700 samples of gut bacteria from 600 healthy babies, 175 mothers, etc. And they were looking at fecal samples at 4, 7, 21 days and infancy. And what did they notice? Well, they looked at vaginal first. And what did they notice? That the microbiome is not from the vaginal bacteria, which was, by the way, was thought to be the case, but in fact is from the mother's gut bacteria. That's what predominates in the first number of weeks and even into infancy. But if the child was born by cesarean, then the bacteria are mostly the bacteria from the hospital environment and mostly skin, which is not what the baby needs. By the time it gets into infancy, you start to see that approximating what happened with the vaginal delivery, but never quite gets there. Oh, and by the way, the interesting thing was that to date, Most women that go through cesarean section are, of course, offered pre-delivery antibiotics to prevent post-op infections. So the baby is not only having the problem of where it came out of, but it's receiving antibiotic doses via the placenta. So that may be causing some of the microbiome differences that we're looking at. Another thing is what happens to that baby when it is being breastfed. And I'm not even going to deal with non-breastfed babies. Well, we know what's in breast milk. We have a very good idea of what's in there. Well, one of the things that you see is here in red. It's human milk oligosaccharides. Okay, HMOs. Why are the HMOs important here? Because the HMOs are prebiotics, not for the child itself, but for the commensal bacteria. That particular type of prebiotic that is produced by the mother's breast milk is there to feed the bacteria, because you want to make sure that those commensal bacteria are going to be able to generate the short chain fatty acids, because that's what they produce. So right from the get-go, the mother and baby know that they've got to keep those bacteria happy. So I want to show you something. This is what I told you a little while ago, but each one of these points being a particular microbiome in an individual. And I want you to see what happens when an infant goes from infancy into the stage about 1,000 days later. So I want you to watch the bouncing ball over the first 1,000 days. And each one of these is a week. This is every week they take feces. And look what's happening. It is slowly but surely going from where it was as an infant. It is beginning to develop the feces of adult life, and then something happens. And what happens? Watch closely. It stops. What is given? Antibiotics. And oh, wow. It goes way up and recovers. Let's do it again. At some point, it goes up and then goes up. It reverts back to infancy and then recovers, and then eventually will reach the level of the adult feces, and that happens after about 840 days. So wait a second. You give antibiotics again and again. Do you think that that may have an effect on how the microbiota are going to affect the bacteria? I would submit to you absolutely yes. Here is some new information. Healthy aging. A microbiome is continually transforming with age, and those who show early signs of frailty as they age have a lot less microbial diversity. Or put it the other way, if you have more diversity as you age, you become less frail over time. Because we know that after the age of 40, there is a distinct microbiome change that begins, and those that age with more diversity are more resilient, etc. They develop higher levels of indols. By the way, those are metabolites from gut microbiota that reduce inflammation. They maintain barrier integrity. And there's a steep decline in Bacteroides, which is very advantageous, since if Bacteroides were to remain at a very high level, they are known to attack the mucus layer, a much thinner layer of an aging individual. And they will trigger an immune response and, of course, set off chronic inflammation. So what's the importance when it comes to diet? Well, we know that diet helps preserve the mucus layer while you're aging. It increases the amount of fiber, and you increase indole production. So you're maintaining that barrier protection. And we know, just for you to know, and you'll have this on your PDF in case you have nothing better to do, keep looking back at that, so you'll know how the gut microbiota affect various types of metabolic disorders. And increased gut permeability, and why is that important in obesity? This is a classic piece of work that was done with rats, obese and lean rats. What did they do? They took mice with obese twins microbiota. So you had a twin, the twin was obese, you took that microbiota, you put it into a cage with mice that contained lean microbiota, the lean co-twins microbiota, and then you co-house them, and what happens? This co-housing of these two types of rats prevented the development of increased body mass and obesity-associated metabolic phenotypes in the obese cage mates. So what does it do? Because they're living with the lean ones, they started to develop the microbiota of the lean rats. So correlated with the invasion of specific members of Bacteroidetes from the lean microbiota, and that was diet dependent. So these findings are revealing that there is a rapid and transmissible and modifiable effect of diet by microbiota interactions. And by the way, if you have increased diversity, that can lead to bigger problems of obesity. So if you take a look at lean individuals, they have more diversity than the obese individuals. Because there's more diversity, here are the lean individuals and here are the obese individuals. This is study after study, which again, we are not going to discuss in any great detail. I'm not even going to bother going through that. What I am going to concentrate on is the metabolic aspect of what the gut system does for us in terms of fermentation, small chain fatty acids, etc. So what about antibiotics? Well, 80% of those are sold in the US for animal production. And we know why. It decreases gastrointestinal infection in cows. It increases the weight of the animals. And to date, 50% of beef, chicken, etc. are still being given these antibiotics. And we know that there are about 35 million pounds in meat and poultry production, almost to date. And that is used in manure fertilization. It gets into the plants, increases their concentration, it gets into food, and on top of that, there's more than 8 million pounds being used to treat human disease. So when you put all of that together, we got a problem. And so the situation today is an increased risk of allergies and food sensitivities, and especially if antibiotics were given in those first 2-3 years of life. It can program the body systems for the remainder of life, and especially the immune system. We do know, by the way, if you go into the gut and into the colon, that when it comes to polysaccharides, humans cannot break them down. They've only got 20 genes that encode for the proteins that break down carbs. So what's breaking down the carbs? Well, we know it's being broken down by bacteria. Bacteroides alone breaks down about, who knows, they have about 260 genes, at least, in Bacteroides that break down polysaccharides. So, and this is important, microbiota are not just helping the body in digesting, they are essential for digestive function. Keep that in mind. Antibiotics in a healthy individual are very important because those commensal bacteria are competing for the oxygen, which keeps pathogens at bay. It keeps them from overloading the system. And because of that, and because of a beautiful mucus layer, and IgA, remember I mentioned that, and the defensins and the other types of antimicrobial peptides, they keep everything balanced. But oh boy, if you give them too many antibiotics, all hell breaks loose. You begin to deplete the ability of commensals to fight those pathogens, and you get an increase in all of the different pathogens, in the toxins, in salmonella, in shigella, you name it. And that's going to cause eventually a systemic dissemination of pathogens into the bloodstream. And so for the last part, the last 10 or 15 minutes, let's just take a look at nutrition. I don't want to spend a lot of time on food sensitivity. Unfortunately, many of our colleagues spend too much time on that and think that the whole game is food sensitivities. It's part of the game, but in many cases, it's a small part of the game. And so doing only food sensitivity testing, I'm just going to throw this out, it's a bit of editorializing, may not be, I suggest, the only thing that you should do. In fact, we know that with food sensitivities, we only know about maybe 17%, 20% of the causes. About 83% is unknown. And we are beginning to realize that it is due mostly to dysbiosis, here we are again with the bacteria, and chronic stress, which we will not discuss in detail this week. Reed and I will discuss that next month. But there is an evolution of the diet, and here we're going to try to bring things together. We talk about the Mediterranean diet and what makes it so interesting and so special. Well, we're going to talk about the start and decline of our diet. 2.5 million years ago, the race begins. The human race appears and it goes through its various stages. And up to about 10,000 years ago, we really had a diet that was fruits and tubers and nuts and meat. We were getting a lot of exercise because we were running away from carnivores that were trying to eat us. And about 10,000 years ago, there's a change from nomadic life to farming life, and agriculture begins to change. And one of the changes, not the only one, but one of the changes is the increasing amount of gluten and gluten-like grains in the diet to this date. And so the diet and the microbiome go through some very dramatic changes. The first is a diversity change in the human microbiome. As I said earlier, it's much less than it was in earlier days and in non-industrialized societies. And that, I submit to you, ladies and gentlemen, is driving many of those modern diseases. I hope I've made that point. Autoimmune, asthma, metabolic syndromes, you name it. And the lowest diversity is in the Western postmodern societies. But then why is it low in all humans? Because of those dietary changes. Because you've gone from nomadic to sedentary agricultural life. Gluten grains become more prevalent. Cooking foods decreases decrease the need for breaking down those plant fibers. And the decrease, by the way, in what we need, which is fiber and fermented foods. That has decreased in our diet. It should be increasing. And of course, the use of the antibiotics. And so there's a suboptimal gut community in modern societies. I'm not going to belabor the point. And how do we improve it? Well, I think what we've been saying is increase the fermented foods, whichever they are. I'm not here to give you the long list. You're better at that than I am. Increase dietary fiber, which is a form of prebiotic. Why? Amongst other things, it's feeding the commensal bacteria. And probiotics. But by the way, and I want you to remember this, if all you're doing is giving probiotics without decreasing inflammation and without fermented foods and the fibers, guess what? It's going to do very little. The body is going to expel it in 24 to 48 hours because microbiome is very effective in its resistance to other colonization. When it does work is if you give probiotics with the fermented foods and the fiber diet. And here is the last part of this. Sleep. Get sufficient sleep. And you want to get, if you can, at least the classic 3 cycles of REM sleep, if not 4. Why? Well, for example, most restriction diets we know are based on an 8-hour window of eating and a 16-hour period of relative fasting. Well, during the sleep cycle, what I'd like you to know, if you don't know it, is that the GI tract initiates emptying waves every 90 minutes. It pushes a wave through the gut, going down to the anus. So it's moving out undigested food. It creates a fluid secretion wave that is moving down distally. And the other thing it does is that if bacteria from the colon during the night are trying to go back into the small intestine, it pushes them back out towards the distal or towards the anal area of the colon. So if you have microbial stress, it's due in part to insufficient cleansing time. And that's why you want high fiber and high fermented foods, which may be a very powerful modulator of the human microbiome and that axis with the immune system. And it's going to be, I think, and many people who are studying this now, a very interesting avenue to combat non-communicable chronic diseases. And I want you to understand that the mucosal environment is a very interesting microenvironment in which, and out of this I want you to remember that the mucus layer has got two layers. There's an outer layer and an inner layer. And that is what's called the mucus microbiome or microbiofilm. You do not want many bacteria to get in too close to these wonderful one-cell layer in enterocytes. You do not want high levels to get in. And a lot of the cells and the bacteria are sitting in this mucus layer, preventing stuff from happening. That is the microbiome geography. And if you eat too frequently, the microbes begin to migrate from the lumen and they start to penetrate the mucosa and they get closer and closer to that cell layer. You want the microbiome in the lumen. You don't want them sitting in high levels in the mucus layer and near the cell wall, because that's going to cause problems. So the dietary interventions that we have, and this I'm giving you for the PDF to increase the effectiveness of the intestinal permeability, is glutamine and all of the effects that it has. And I'm giving you this so you'll have it on the PDF, its role in suppressing pro-inflammation, etc. And glutamine, by the way, has a tremendous amount of functions. The most important are modulating the tight junctions and promoting SIgA secretion. The flavonoids, the phytonutrients, of which you know about many of them, also extremely important. One of the most important is berberine, and I think that you know about that. And last but not least are the probiotics, which, as I told you earlier, are extremely important in balancing the microbiome, which will benefit immune function and will improve all of the gut function in general. So here's what I want to do in summary. This is an important slide, and I want to spend a few moments on this. What are the mechanisms then of chronic inflammatory disease development? The influences of prenatal, perinatal, and postnatal. That's going to develop all of the issues which we discussed earlier. That is going to change the microbiome composition. That is then going to alter gut permeability. It's going to allow an increase in antigen trafficking through the gut wall, which is going to increase the immune response, which can lead to abnormal immune response, which then is going to create a very dysbiotic and then pro-inflammatory cytokine development. And if you do too many developments of antibodies, you are going to increase low-grade chronic inflammation, and it's going to lead to chronic inflammation. And the microbiome is important. And why are the first 2 or 3 years so important? If you had vaginal delivery, proper nutrition, remember the first 2 to 3 years, low infection rates, low amount of antibiotics, you are going to create a very healthy microbiome composition. And that's going to lead to a balanced microbiome. Whereas if you have a C-section, improper nutrition, multiple infections, multiple antibiotics, that is going to lead to dysbiotic behavior. And in the case of a balanced microbiome, you have a high threshold for GALT. Why is the immune system at a high threshold? Think about it. 2 million years ago, the only thing humans needed to worry about was they were going to be eaten by a carnivore or they were going to have infection. And if they had infection, that was the only time they wanted to send out the big guns of immunity to treat that infection. Otherwise, they did not want those inflammatory, that innate immune system to be functioning. So it has a high threshold. You don't want it to work normally. You want energy. So if you had a positive genetic predisposition, a good genetic predisposition with a high threshold for GALT, guess what? You have a good state of health. But if you are unfortunate enough to have dysbiosis with a genetic predisposition, you end up with inappropriately low threshold for GALT. And you're developing inflammatory problems constantly. And that leads to chronic disease and various types of chronic inflammatory problems. So a lot of people would say, so wow, that means the microbiome is the answer. Let's invest in microbiome companies. Let's just focus on the microbiome. I would say, whoa, time out. Not so fast. Is the microbiome a part of the problem? Yes, it is. Yes, it is. Is it in fact going to be a very important part? Yes. But we only know about what's going on with 3, 4, or 5% of the species of bacteria that are there. We know the microbiome is important, but we don't know how to use it as a treatment modality in a predictable way yet. Will we be able to in the future? Absolutely certain we will. But can we do it now? We don't even know who's there, number one. We don't even know how it really functions. It's not the one species. They function together. We're not even sure what those populations are. And we are not sure about the ecology. We don't know how they're interacting. So I would submit to you, we're still in the infancy. Should we do microbiome testing? Yes. But is that the way in which, should that be the fundamental test we do? I would submit to you, no. We're not quite there. But is it a part of what you have? We call it the cornucopia of plenty. Is it in your armamentarium? Do you have it in your tool chest? Yes. And what is the state of the microbiome right now for clinical application? Well, this is adapted, and you can read about it in the classification of microorganisms starts with domain to the kingdom to the phylum and down through class and order and family and genus all the way down to species. And you can look at the various types that would deal with that. It's almost like saying the domain is the universe, and then you work through the Milky Way and the solar system until you eventually use, Reed, this was for you, San Diego.
Love it.
Where are we now? You know where we are? We're at the solar system. We don't really know predictably what's going on planet Earth, let alone knowing what's going on in California or San Diego. So are we ready for prime time yet with the microbiome? Not yet, but it's getting there. And I'm so happy to say it's getting there. And remember, our genetic complexity depends on that microbiome. By ourselves, we don't even have as many genes as some worms that are in the earth. So how are we able to do all these things? Because we have inherited the 3 million or more genes of the microbiome. And together we are flourishing. Together we are doing this. So we have to thank the microbiome for that, including for keeping us alive. Because in our cells, the mitochondria that keep us alive, that give us our energy, guess what? The cells millions of years ago, many millions of years ago, brought in bacteria into the cell, and those bacteria are the mitochondrion. So guess what? The mitochondrion is an integral part of our cell, and it is what gives us our energy. It couldn't get better. So the future is multi-omic analysis. Microbiome, metabolome, nutrigenome, I'm going to say them all, transcriptome, the epigenome, the genome, the proteome, which together allow us to integrate the data that gives us the multi-omic portrait. And that leads to the exposome, which is the totality of all of these external environmental exposures. And that's giving us the ability to know what happens at both the cell and the organ level. And that's what Reed discusses, to know what is the underlying root, to take us right back to what you discussed right in the beginning. You want to know dress. You want to know what's underlying it. And that's what I discuss in the summers, which I have in Canada, the summers with all of the winter. I'm joking. This is in the middle of our winter, so we love our summer, which is coming to an end. Thank you for giving me the time. I know we started a little bit late, and thank you for your attention.